The term linked genes refers to genes that are located on the same piece of DNA. And since they're on the same piece of DNA, that really means that they're on the same chromosome. And so this linked part refers to the fact that they are physically connected to each other. And so as we've drawn it here, I've got this chromosome that's got several different genes. I've drawn 5 in this case that are all located on that same chromosome. And really, when we're talking about real human chromosomes, you have hundreds of genes that are located on there. So if you were just to randomly choose 2, it's very possible that they could be located on the same chromosome. Well, when we start talking about inheritance patterns and we're dealing with linked genes do not assort independently. In other words, they tend to be inherited together. So that is the tendency. So the tendency is to stick together, which is going to really throw off the inheritance patterns. So when this chromosome gets distributed into the gametes, whatever's on that chromosome is going to end up in the same place. Now, we do sometimes get separation of the genes when they're located on the same chromosome, and the process for that would be crossing over. So if we do happen to have crossing over will separate linked genes. Now, the probability of crossing over actually occurring between 2 genes that are on the same chromosome is going to be dependent on really how far apart those genes happen to be. So here we have an example where we're looking at 2 homologous chromosomes inside of an organism. And in this particular organism, the genotype would be heterozygous for A because we've got a big A and a lowercase a. And it's also going to be heterozygous for B because you have one of each. Now, if these genes happen to be located on those same homologous chromosomes, as is the case here, we would expect that most of the time, the versions that are next to each other, so in other words, these 2 capital versions, since they're on the same chromosome or the same piece of DNA, they're going to tend to be inherited together. Or in other words, if we just draw directly down here what we would expect to get in our 4 chromosomes when they separate into 4 different gametes, we would expect that this is what we're going to have. So we're going to have chromosomes that have both of the capital versions, and we're going to have chromosomes that have both of the lowercase versions. And again, those would get separated into 4 different gametes so 4 different eggs or 4 different sperm. However, if a crossover event occurs, then we're going to get some recombinant chromosomes. So if crossing over occurs, and more than just if it occurs, but if it occurs between the 2 genes we are studying, then we get new combinations. So in other words, we're going to see these 2 genes or alleles, they're going to be separating from each other. So if we look at this, if this chromosome, if this one here overlapped this one, so if they broke right there and they swapped pieces with each other, what we would end up with is going to be that those versions get switched. So if we were to draw that on here, we're now going to see a little a over here and a big A over here. And so those are the recombinant chromosomes. And note how that is going to change what we get down here at the bottom. So at the bottom, we would then have these new recombinant chromosomes. Again, new combinations compared to what we had before. So this one and this one, those would be the recombinant. And really, they are the result of crossing over happening. They tend to stick together, these 2 particular genes, because they're on the same piece of DNA, and we call them linked. But if a crossover happens between them, then they're going to separate and we get the recombinant chromosomes. If they're located very close together on the same chromosome, then we don't get very much crossing over between them. If they're located further apart, then we have a greater chance that there will be a crossover there, and then we would expect the frequency of these to actually increase and go up. Overall, when we do have linked genes are going to alter the expected frequencies. So the expected probabilities when we look at the results of our crosses. So if you do find that, you were expecting to get 50% of one thing and 50% of another, and if that ratio was drastically altered, then that would be a clue that the two genes that you're looking at are going to be linked to each other.